Now we'll turn our attention to tumor suppressor genes, with the subclass of those being DNA repair genes. So, like the name implies, the goal is to suppress the growth of tumors or inhibit cell growth. When they are mutated, they're no longer able to do their job. They're no longer able to stop the cells from dividing, hence stopping cell growth. Therefore, this is a loss of function mutation. They do not do their job anymore. So the two-hit hypothesis was identified in tumor suppressors in retinoblastoma, and this is in 1971 by Alfred Knudsen. So he said that it takes two hits, two mutations, making this a recessive disease. So if you inherit it, you already are born with one hit, okay? So you have one mutation. You only need to acquire one other mutation to lead to the growth of tumor. Because you are born with one and only need to acquire one more, these tend to show up earlier in life. Whereas if it's sporadic, you're born with two normal copies, and now you have to have two mutations over your lifetime that leads to cancer. Because this is such a rare event and you need these two mutations in the same gene, it takes longer to accumulate. So these tend to occur later in life. But the idea is that you have to mutate both copies of a tumor suppressor in order for cancer to develop. So they have identified tumor suppressor genes. And remember, they can have two functions. One, negatively regulate cell division, or two, work in DNA repair or genome integrity. And so RB was the one identified in retinoblastoma. Since then, they have identified P16, NF1, APC, and then P53 and the BRCA1 and 2. When you hear BRCA1 and 2, you should know these are those breast cancer genes that we've talked about before. So this P53 was the second tumor suppressor gene identified, but they found out this is like a master tumor suppressor. It has shown to be mutated in about 50% of all human cancers. And these tumors can show up in a variety of organs: lung, breast, esophagus, bladder, brain, liver, sarcomas, lymphomas, leukemias, okay? So over 50% of all cancers and different cells. Now what happens is P53's job is to determine if a cell has DNA damage, okay? So if a cell has DNA damage, you're going to see an increase in the expression of P53, and this is usually in DNA double-stranded breaks. So P53 itself is a transcription factor. So remember, it has two binding sites, the one that binds to the DNA and then the one that activates it. So if damage is detected, normally P53 can do one of three things, okay? So it can activate genes that are involved in DNA repair. It can stop the cell cycle from progressing so that the cell can repair its DNA so that we're not passing on these mutations in every subsequent cell. It can also tell the cell to apoptose. So remember that cell death that was mentioned in another one, okay? And this is through the release of caspases. Now, here we go looking at this P53 again and all the different things that it can play a role in. So remember, this is a master tumor suppressor. When there's a loss of function, P53 cannot do its job, okay? So it cannot cause DNA repair. It cannot stop the cell cycle from progressing. It cannot cause apoptosis. So what happens? We're no longer able to control cell division. We're no longer able to ensure genome integrity. And that's in the form of one, stopping those mutations or preventing those cells from surviving. So these two are, we can put a mutation in those checkpoint proteins or those DNA repair proteins as well. So those checkpoint proteins we have a role in cyclins and CDKS. So these cyclins CDKs, the cell regulates how much or how many are produced. Okay, so we are looking at how much of one do we have in relation to another. And their expression changes as we go through different parts of the cell cycle, but we rely on those to get by those checkpoints. Okay, So if all things are good and we can advance, then they'll be degraded, the other one will increase, and we'll do this over and over. So those CDK cyclins are important. Now, if you mutate them, now you no longer are able to regulate the cell cycle in the same way. So now we can't say if there's damaged DNA. We can't stop the cell from progressing if there's damaged DNA. We can't activate apoptosis if there's damaged DNA. Okay? We're just going to let it just keep going and going. If we mutate those DNA repair genes, now we cannot repair that DNA. So all those things we talked about, we can't do direct reversal of thymine dimers. We can't cause base excision repair. We can't do homologous recombination repair here. So now we can't fix those errors. Now those errors are able to progress and be passed on to every daughter cell that comes from that cell. So remember, we looked at those mutations in the DNA repair genes, and we said they have an increase of cancer associated with them because we can't repair those. And a hallmark of cancer is lots and lots of DNA damage. So when we damage that DNA, we're looking at mutations because remember that cancer is a genetic disease. So we can directly put a mutation in that tumor suppressor, causing it not to work anymore. So what are those mutations? It can be a missense. It can be a nonsense. Okay? Perhaps we might identify a frame shift, or we can cause aneuploidy. So remember aneuploidy, where you have either a gain or loss of chromosomes. Well, if you lose a chromosome that contains a tumor suppressor, that's like a hit. You don't have that chromosome. You're relying on one chromosome. It's easier to mutate one chromosome. And now I want you also to bear in mind that cancer takes multiple changes to lead to malignancy. It's multiple ones over and over again. So what happens is you can get mutations in tumor suppressors and oncogenes, and a couple tumor suppressors and a couple oncogenes, and tumor suppressors, oncogenes, those DNA repair, all in one. So it's usually not going to be just, one mutation. Sometimes it might not be two mutations depending on the type of cancer it is. So if we take, for an example, colorectal cancer. So you have this normal, then you lose a tumor suppressor. So you start to get this growth, okay? But this is benign at first. It can't invade, it can't metastasize. But then you get another mutation. Now it gets bigger, and another mutation. Now it's able to invade and metastasize, okay? So it's mutation after mutation after mutation. But also remember that because this cell is now rapidly dividing, it can incur those mutations because it's dividing more than it would. And so now the more cell division you have, the more likely you are to incur a mutation. So we talked about some cancers having a genetic basis. So we said 5 to 10 of our cancers are inherited. Well, here are some tumor suppressors that have been associated with genetic diseases and leading to cancer. Now, it doesn't mean if you have a mutation in one of these that you're going to get cancer. It means you have a predisposition because now remember, you're born with one hit. So you only need one more hit for that cancer to develop, okay? So you have an increased risk compared to the general pop. So what does it mean? So you have people, you get that one hit. You're more likely to develop cancer, but it's not guaranteed. So now what happens is you have inherited one mutation. You need to occur one more. Now you need to get an induced or spontaneous mutation to occur. Well, you get this loss of heterozygosity. Now you have 0 copies that work, okay? So here you're a heterozygote, one mutant, one normal. You lose that normal, you have a loss of heterozygosity, you don't have any normal. Now you're likely to get cancer, okay? So that's our lecture on tumor suppressors. Questions, comments, concerns?